Hey guys, it's Ryan, and this video we're going to look at the auditory pathway for processing hearing. So let's start out with a sound wave, and a sound wave is just a vibration, typically of air, of oscillating waves of compression and rarefaction. And these oscillations are going to enter in to a conveniently shaped external auditory meatus that focuses the sound wave until it contacts the tympanic membrane, which is at the boundary of the outer and middle ear. And the tympanic membrane will bow in and out in perfect sync with the sound wave. So when you hit a compression, the tympanic membrane will bow in, and then when you go down at the bottom of this wave to a rarefaction, the tympanic membrane will bow out. And then when there's no more sound, it'll go back to resting position. Now there's one problem, because usually we have sound coming in from air, and then we're going to have to transition to fluid in the cochlea. So there's something called impedance here, because we have two different media, and there's a lot more resistance in fluid. So we have these middle ear bones to help compensate for the impedance mismatch. So this is the malleus, the incus, and the stapes. And one cool feature is that the tensor tympani muscle attaches to the malleus, and the stapedius muscle attaches to the stapes. And if there's a very loud, sustained sound, those muscles will tense up and dampen the sound to protect these delicate ear bones, or ear ossicles. So when the sound reaches the tympanic membrane, it'll activate this lever system of the middle ear bones, and then this stapes will contact the oval window, which is a membrane on this part of the cochlea, which is this whole sort of snail-shaped apparatus. And there's liquid in this outer compartment, which is called the perilymph. And the perilymph is very similar in composition to cerebrospinal fluid. So it'll have ions, proteins, and glucose. And what happens is this fluid will, or these waves, these vibration waves, will travel through this fluid. And the apex of the cochlea is called the helicotrema. And then the waves will come around this side, and eventually pressure will be relieved through the round window at the bottom. So I should mention that these compartments are given different names. The top one is called the scala vestibuli, or media, and then the bottom one's called the scala tympani. A lot of different terminologies here, but so it can sometimes be hard to keep it all straight. But that's all that. And then we do have a different fluid in this region that we didn't talk about, which is called the cochlear duct, and it contains endolymph, which makes sense because endo means inside, and the perilymph is around the cochlear duct. So the endolymph has an unusually high concentration of potassium ions, which will come into play later when we talk about the electrical component of this pathway. So now what happens when we talked about the waves sort of traveling through this liquid, this perilymph liquid, and it can be relieved through the round window. But some of the waves will enter in or try to work its way into the cochlear duct. But there's a lot of bone surrounding this cochlear duct, so there's only really one way for the waves to penetrate in, and that's through the basilar membrane. And I have this little pink line running through the bottom of the cochlear duct, and if we zoom in to, say, a region right around here of the cochlear duct, we're going to get a nice zoom in of the organ of Corti. So that's what this is. And so we have our cochlear duct, and here's sort of what it looks like inside. Here's the organ of Corti, and there's some more terminologies we should get familiar with. The basilar membrane at the bottom, and then this is the tectorial membrane. And so the vibration waves, the sound waves, will activate this basilar membrane, which then causes these different vibrations in the hair cells. And that's going to be these orange cells here, and these are really our most important part of this whole pathway, because they're technically the receptor cells. And the hair cells are organized in a very particular way. There's one row of inner hair cells, and you can imagine this is just sort of one cross section of the cochlear duct. So we just kind of took one cross section, but these hair cells extend far back. There's a whole row of them, long row going all the way along this cochlear duct. And then there are 3 rows of outer hair cells, and their functions differentiate slightly. So I think it's a little important to point it out. Anatomically, they're very similar. They have these little hairs sticking up that are stuck in the tectorial membrane, and that sort of fixes them from the apical side. Then from the basilar side, they're attached to cells, sort of piles of cells on top of the basilar membrane that vibrate with the sound waves. So that's what causes these hair cells to move. The inner hair cells, that row is more inclined to help with the sound recognition, whereas these the 3 rows of outer cells are special for mammals, and I think a couple of bird species. It's more for fine-tuning of sound, for frequency discrimination. That's just kind of like a special add-on. So let's hone in on the inner hair cells. So we isolated one here, just zoomed in a little bit further, and these hairs are pretty complex structures called stereocilia, which is a nice name because stereo you think of sound, and cilia you think of hair. So there are these stereocilia, and the largest one is called the kino or kinocilium. And because the basilar membrane is vibrating, these stereocilia that are attached to the tectorial membrane are going to move as the hair cell moves. So they're going to kind of slide back and forth depending on if the basilar membrane is activated or not. And there are these little strings attaching the stereocilia called tip links. So they just link the tips of the stereocilia together. And those will either be stretched or relaxed depending on the direction of the stereocilia. So if they're leaning up against the tallest one, which is the kinocilium, then the tip links will be stretched fully. And that means that this hair cell will be turned on. And if the stereocilia are facing the opposite direction of my picture, away from the kinocilium, then the tip links are relaxed and it's turned off. So I kind of think of it like a light switch. This you can turn it on if you push it in one direction, turn it off if you push it in another direction. And it's actually, this is really fast. They're turning on and off in perfect sync with the original sound wave. So this is all completely in sync. Okay, so when the tip lengths are stretched, we said that the hair cells turned on. And what we mean by that is that these little channels, they're non-specific cation channels. I have them in purple here. And they are mechanically stretched open. They're mechanically gated ion channels that allow for the passage of ions when the hair cells turn on. And if you'll remember, the endolymph that we talked about, which bathes which is inside the cochlear duct, bathes these hair cells. And so when the cation channel is open and there's a ton of potassium ions out there, they're going to rush in. So usually this is opposite of what we're used to. We're used to sodium rushing into the cell, but this time it's potassium. But the effect's the same. It's still a depolarization because the inside of the cell is becoming more positive. Now, this isn't an action potential. This is just a receptor potential, which is a form of graded potential. And that graded receptor potential will flood the cell. This will become relatively more positive along the membrane. And then this will cause the influx of calcium that we're used to, which will cause exocytosis of vesicles with neurotransmitter. And then the neurotransmitter will activate sensory nerve endings of an afferent neuron. And this will be part of the cochlear nerve, which is a convenient term for something that is coming from the cochlea. And this is just part of the vestibulocochlear nerve, which is cranial nerve 8. So it's just one of the components of that big cranial nerve. All right, so that's pretty much all the physiology. Now just for the neuro part. This is the cochlear duct. It kind of wraps around. It's coming up towards the brainstem. And the first synapse is in the cochlear nucleus, which is another great name. The cochlear nerve goes to the cochlear nucleus, and this is in the medulla. And then this isn't really well understood, but there are branches that go right and left. And there's sort of more that go one direction than the other direction. And that's to help position the sound, the source of sound, so we can know if there's a stronger signal on the right side that the sound is coming from the right side of our body. And so these branches will go up through the pons. Some will synapse in the pons. There's a whole lot of synapses going on, but the most important one is in the inferior colliculus. And there are two of these right underneath the superior colliculi, and this is for auditory processing in the midbrain. And the superior ones are for vision. So synapse is in the inferior colliculus, then this will go up to the thalamus, and then eventually we will get to the auditory cortex, which is located in the temporal lobe. Auditory cortex. Now, there are a couple, just two things I want to point out that are pretty cool. One is that the basilar membrane is very particularly organized. It's stiff and narrow where it is by the middle bones at the base here, and then as it moves towards the helicotrema, it gets sort of floppier and wider, and that makes it so that it's extremely well tuned, and it's called tonotopic organization, because depending on the pitch of the sound or the tone of the sound, it's going to activate different parts of this basilar membrane. So when we took a cross-section here, and we showed this cross-section, this hair cell will only be activated by a very certain frequency of sound. So here at the base, we're going to have high frequencies, and then we're going to have low frequencies here. So it's sort of like, you can think of it like a piano. The smaller notes are the higher pitches. So here, the smaller, the narrower part of the basilar membrane is responsible for higher pitches, whereas the wider part is for lower. And then that maps onto different parts of the auditory cortex. So it's almost like a labeled lines hypothesis, where you have like all of this mapping at different portions, and then particular hair cells will send up particular neurons that'll go to particular parts of the auditory cortex, depending on frequency. But then how does the body know, differentiate loudness? And that's through the amount of neurotransmitter released. So a louder sound will turn this hair cell on more powerfully, more frequently, and then more neurotransmitter will be released, which will cause an increase in the frequency of action potentials, which again is the primary way for the PNS to communicate to the CNS the intensity of a stimulus. Alright, I know that was a lot, but I hope it was helpful, and I'll see you all next time. Thanks for watching.